Okay, that should be streaming. Is it actually streaming? Maybe it takes a second?
I need to, my cat's going a little wild. I think she wants to get
No,
out
you're
and doesn't
fine.
know how to do audio yet. Oh, cool. I'll
Yeah,
be
you're
right
good.
back.
Trying to figure out what's
Thank you,
going on
thank
with
you.
this. Hmm. Ah, there it is. Aha. Okay, let's copy that. Okay, there we go. Things are figured out, so let's open up the project.
Woohoo!
MDL 15? I don't know if I've actually looked at any I've looked at a couple sequences from this plate, I think. Maybe not. I haven't been doing as much OMDL stuff as I probably should.
Oh, yeah.
But I've just been busy.
Absolutely.
Insanely stinking busy.
Well, that's good.
Eh,
Probably. Fair enough.
Defibrillati? What? I don't know what that is. I'll find out in a second. Oh, it's Cornarius. I guess I should not have been surprised. Who is this from? Melissa Dubois. I don't know this person. Interesting. Probably.
When it says section, what does that entail?
So it's just kind of like a taxonomic rank, like between species and or like genus and species, right? It's just like kind of like
Oh,
a sub rank. So if I go
so-
to it on the actual page for it and look at the taxonomy, so
oh,
it's in
oh,
subgenus Mixaceum in section Defibrillati. It's just kind of a way to
interesting.
further like further
Like
separate stuff
delineate.
out, because like some of these genuses have hundreds and hundreds and hundreds of species, and the sections kind
Sure.
of help sort them in a way that's it's useful, because like when you look at like
Oh,
Amanita section Phalloideae is full of things that will melt your liver, and Amanita section Caesareae is full of delicious things you can eat, right?
wow.
So
Okay.
ideally, it should be like a monophyletic group inside a genus that is also monophyletic, but not everything is monophyletic because taxonomy, especially with fungi, is a mess. Um,
Monophyletic. Man, there's so many words.
do you know what monophyletic?
No.
Okay, so basically, it just means everything under like in a clade is going to descend from like a single common ancestor, right? So like
Okay.
oh
Okay.
gosh, I don't how would I describe this? So polyphyletic would be like maybe I'm not as good as explaining cladistics.
You're fine. I can also like just do some research.
Yeah, okay. It's like basically polyphyly is like if the group is okay, here's what I was looking for. Here we go. Here we go.
Ah,
We've got
okay.
like Wikipedia has some good graphics, right?
Sweet.
So here we have this phylogenetic tree, right? Blue is monophyletic because they all share a common ancestor.
Oh, okay, okay. Gotcha.
If they are here's a better example. So polyphyly would be like something like this, where you try to take two groups that aren't descended from a single common ancestor, right? Because, well, they are, but lemurs are also descended from that same common ancestor, right? So you can't group lorises and tarsiers in this example without also grouping the lemurs together, because the lemurs are more closely related to the lorises than the tarsiers are to the lorises.
Interesting.
And then this
Okay,
is, yeah. And then
okay, cool.
paraphily is like when you have a group like this, and there's other things that share a common ancestor, but you're only grouping that group together. So a good example is how some people want to say that humans aren't apes or aren't monkeys.
Sure.
Because, so I only use this as an example because I watch like a bunch of primatologists on YouTube.
All
But
right.
like,
Nice.
so we're great apes, right? We're descended from, uh, our most closely related species is the genus Pan, which is chimps and bonobos. And then the next most closely related one is like gorillas. The next one is orangutans, and then the next one is gibbons, right? So like that's the monophyletic like ape clan, right? Apes, and then you take out gibbons, and everything after that is great apes. But great apes and apes in general are more closely related to old world monkeys than old world monkeys are to new world monkeys. So necessarily, if you want to group old world and new world monkeys in a monophyletic clade, you have to include great apes. Otherwise, it's paraphyletic.
Interesting.
Does that make sense?
Okay.
I if
Um,
you want better explained videos on this topic, I can link you some stuff.
okay, yeah, that would be cool. Yeah.
Yeah, I can.
Thank you,
Yeah,
appreciate it.
look up Clint's reptiles on YouTube. He does a lot of like videos on like cladistics of different groups, and then look up. There's a guy, but this guy is one of like the best science communicators I've seen on like
Oh,
anything.
wonderful.
But he has this playlist right here, which is just kind of like going over. Oh, that was loud. Evolutionary concepts, right?
The light of evolution,
Mm-hmm.
or what is?
Yeah,
Oh, cool.
it's really good. Like he's got like he's like a bioanthropologist that's going for like his second master's. It's dude is ridiculously smart.
Lovely, lovely. Well, thank you. Appreciate it.
Like I'm pretty sure this guy would run circles around me if that like,
Well, that's
insane.
like his concentration.
Yeah, but no, insanely smart person, right?
Nice,
Anyway,
nice.
uh,
So,
did that help?
mhm.
Sweet.
Oh, it told me there's so much more that I need to know. As
Yeah,
always, right?
I go down a lot of science rabbit holes. A lot. Like I tend to be watching like educational videos while I'm doing this sort of stuff, or watching
Oh, okay.
watching you know atheist content and trying to prove to myself that there's sane people in the world that aren't gonna.
Oh my.
Hi, cat. This is one of my cats. He wants friends. He wants pets.
Hi,
Oh,
kitty.
oh,
What's that kitty's name?
this is Samson. He does not live up.
Samson. Hi, Samson.
He's a fraidy cat. He does not live up to his name. It's fine. Okie dokie. What do we have? We have
Oh,
very few close matches. I'm definitely gonna have to look at this one. I don't know why I say that. I look at basically everyone unless it's like a freaking turkey tail.
that's understandable. You're
Yeah,
doing your due diligence.
well, I mean, not everyone's quite so specific about it, but I'm just I like looking at the sequences a lot, and I've just it even the way I look at it is even sometimes if I'm looking at stuff that um is common, it's still practice. So
Oh, absolutely.
probably should make this a little smaller. There we go.
Well, I had a question about the LSU primer we were looking at last night, and
Yes.
that it was backwards. So if we had put that
Yes.
forwards, would we been able to not do the reverse complement, or would that not work out? Like,
Um
if that question
it's
makes sense.
it's just easier. It's just easier just to you're gonna have to reverse something. So like we have the conserve sequences document that Hart was so
Mm.
kind to provide us. Um,
Yeah.
and so this is the
That's
the LR5 is what he's been using, and
okay.
it's just so if you, when you're using agat kata for
Mhm.
ITS, you just know that the
Yeah.
other group of sequences is what needs reverse complemented. If you use this for LSU, you know that this is the group of sequences that needs complemented, reverse complemented, right? Either
If you were
way,
to
you're
if you were
reversing.
to reverse that, would it be like
I mean, you could
this?
reverse complement this if you wanted to, and it would find the same
Okay.
the forward theoretically.
I
I
was
don't
just wondering.
Yeah, I don't see why that wouldn't work. Like you would just have to reverse complement that,
Okay.
but
Cool.
it's
Cool. All right.
yeah. I, yeah.
That works just to do it that way. I was just wondering
Yeah.
if
Yeah, if you
that
were
was actually
it should work if you reverse
legit.
complement it, yeah. Cat, what is
Okay.
wrong with you? It's like I get on
Oh,
if I get on a video chat and they're immediately like, "Oh, she's talking." Like we must bother her.
our cats are the same way. We get on a video, rewatch TV or something, and they're like, "Hey, no, no screams.
Well,
We
he's
don't
also
allow."
their dinner time is also in about like 30 minutes, so that's when they start to
Ah,
really
mhm.
bother me. They're like, "Oh, it's food. Feed us. Fill our gullets." Okay.
Booties.
Alrighty. So I already aligned that. Can you get comfortable? Oh my gosh, what did I do? Um. He scratched his
What?
ear. He'll occasionally like get itchy ears and scratch the heck out of his ears and make that noise and then run away.
Oh, kitties.
He's like, I don't know. He's like, now he's singing me the song of his people. Can you hear that?
No.
That's probably for the best.
Oh
Oh my
dear.
god. Like
Cats are such amazing beings.
he's like
Also,
12 years old too. Like he's not a young cat, so he's definitely exhibiting some old man tendencies.
yeah. Oh,
Okay, now this is aligned.
little loves.
He's a good boy. Cats are weird.
Cats are so
Here
weird.
we go.
How many cats do you have?
I have 3. That's probably too many for the size of my house, but it is what it is.
Yeah.
All right. Oh yeah, so that's definitely 1. That's probably good. All right. I could not believe. Oh, I think you left before this. One of our people was using just his like touch on his laptop, and I'm like, I felt so bad. I couldn't. I tried doing Eugene with my laptop with just um
Oh,
with just a touchpad, and I can't do it.
just a touchpad.
Yeah,
Huh.
like a laptop touchpad. Touch
Is
pad. This is not run over.
in,
I don't
instead of using a mouse or
know.
what?
Yeah, I don't
Oh,
think
I
you.
don't have a mouse.
How do you do it?
I'm I'm used to using a touchpad, so that's how I do it. So that's probably
I can't use a touchpad.
I never use a mouse. If I used a mouse, I would probably be the same way, but
Mhm.
since I never use a mouse,
That's
I've just
fair.
gotten used to it.
That's fair.
Yeah. Yeah, yeah.
All right, that's edited. There's no LSU. Um, I guess we'll see how long that's gonna take. So normally what ends up happening is when I'm going through this, like I'll edit it, blast it, and then just bookmark it, and then go to the
Okay,
next one while I wait for that blast to finish because Micromap sometimes can be a little slow. So
absolutely.
obviously I wasn't doing this in like, you know, uh example stuff in like the zooms, but Amanita porphyria. Go away. Interesting.
Ooh, booty. You know why there's been so many blast searches on all of these?
So this it's a function of how Micromap works, right?
Okay.
So when if it gets uploaded and it's the wrong direction, it'll blast that and then it'll blast it again in the forward read. But also, if any are uploaded with like the same sequence, it's going to connect blasts to that sequence. So
This
Micromap doesn't like the Micromap, it seeds it off your like your sequence in your INAT observation, but the way Steven has it set up is any other blasts that are to that same sequence will also get connected to any sequence that have that same like nucleotide. It's I'm assuming that he does it like that because like these don't update, right? Hi. So these don't update, but doing it this way means that any I can't think of where my brain is going. It means that like every time a new one gets blasted, you don't have to go digging for it or you don't have to reblast it. So theoretically, if this was like a year-old sequence or something that I was looking at and it's been blasted up again, it means I'll have the most recent blast of that sequence and I can look at the most recent data. Because otherwise, I might be looking at a year-old sequence and I then have to like go manually reblast it because there hasn't been a sequence a blast done in a year, and there's probably more information that it isn't in
so
that blast.
if there's something similar or something with the same sequence, that's when it will put the additional like
Mhm.
put the blast in
Yeah.
another like
Well,
and
so like for example, like so here the top ones are from
okay,
Steven and not for Kyle, even though this is Kyle's sequence. That means Steven
okay,
uploaded something after Kyle uploaded this that's
okay.
matching this sequence, and so it's just going to show the most recent blast up top.
Is it would that be a 100% match then?
Basically, but
So if you were looking at the blast results themselves, then you would see the ones you would probably if you had blasts that were populated based upon another one, then you would have 100% result, or am I missing something?
basically Micromap doesn't connect the it 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 doesn't restrict. Sorry, I'm reading what's going on in chat here.
I am too. I'm like, what is happening over here?
um Drop the DOI. I'd be interested in reading the paper. Yeah. Um okay. Yeah, drop the DOI. I'll take a look at it. Um I'm trying. It's hard to explain. I honestly don't think I've managed to explain this in a way that Hart has managed to figure. Like maybe he has, but
You're good.
basically, this sequence
Mhm.
is going to be shared by like other species, other specimens of this species, right? So you're
Sure.
gonna have the
Yeah.
same basic ITS.
Mhm.
So if another specimen of that species gets updated, like say we're looking at a turkey tail and then someone else blasts another turkey tail, and of course they have the
Mhm.
same sequence because they're both turkey tails. Um,
Sure.
that new blast will get connected to every single sequence of turkey tails that matches that sequence so that there's
Interest.
always the freshest blast available. The
That's kind of really cool. Does it
mm,
need to be, can it have like some nucleotides that are different, or does it need to be 100%, do you
I
know, in order
haven't
for it?
dug into it that much. That might be a Steven question. Yeah.
That's really a
Yeah.
cool way of doing it.
Yeah.
Okay.
The one thing
Okay.
is if you do that, so I'm looking at INAT 15308 this one, right? It's not
Yeah.
going to show up at the top of the list. The one that shows up at the top of the list is the seed
Uh,
for it,
for
right?
what?
Yeah. So if you do look at one that isn't the one that's like the one that was seeded off of yours, um,
Uh-huh. Whoa. Okay, this is such good
mhm,
information.
yeah,
Thank you. Wow. All
mhm.
right.
So I am actually probably gonna look at this one because it's the most recent one, and I don't feel like digging for it. But I do have to keep in mind that mine is right here, right? It
Absolutely.
actually has a fair amount of matches. So let's look
Yes, it
at
does.
this. You'll have to forgive me, the mind. I'm very unsure of what you mean by a biohacker form. Okay, so it's not a published paper. It's not peer reviewed. Okay. um well, right, but surely if you're the father of fungal computing, you would have published your paper in a peer reviewed source that can then be referenced in perpetuity. I did not align this yet. What is happening here? Did I mess this up? I messed it up. Getting distracted.
Fair enough. All
I
right.
mean, there is still a way to work with academic scientists without being in academia. Not everyone in academia is full of themselves. I got PhDs on my friends list. I know of several free to publish mycology papers. Are so uh so we're talking about fungal sequence validation. just just saying. So okay. So looking at this one, by the way, April, this is probably one where I am not going to accept this ambiguity, right? Because it looks like we kind of have a little bit of a frame switch situation happening. Like we have this a little T duplication here. We've got a cup like this could be a hollow polymer repeat happening with this extra A. Like it's only the only reason it even popped up past my filter is because this is only a 22 read sequence. Yeah, and hey, that's why we're doing this. You know, there's not really a database for fungal comparisons in as far as DNA goes, and someone's got to start doing it. So yeah, but I I mean I can say from my experience that we work with both you know amateur mycologists and professionals, and everyone that I talk to is they're all very collaborative. So possibly the community has improved since last time you interacted much with it. But
Yeah, I just joined the community and I've
mhm,
been accepted with open arms. If you could post some link to your research, I'm sure that some folks would be interested in looking at it, the mind.
mhm. Okay. Yay, thank you. Um, that's the hope. All right. So, sequence is good. We don't have to do any edits. So this has a lot of matches to Amanita solanolens.
So you said you weren't going to accept the ambiguity, is that what you
No.
said?
Yeah, it looks
So
like it's just noise because that's a really low RIC. Not really. I mean, it's 22, but like we've been getting some really clean breeds back from like sub 30 stuff. But since there looks like there might be a hollow polymer repeat going on, there's like a little bit of a frame shift in a couple of the reads. Like I don't think that's a real ambiguity, right? Like if it was closer to 50-50, that'd be one thing, but it's like barely knocked under my like 70% filter that I have. Um so I'm just
okay.
gonna not worry about that one. Plus, it's got all these 100% matches. This seems like it's something that is like not
It
very
does.
like there's not it's this is not a species that has a lot of like variation, right? We got an N down
Right.
here. This all this means is someone probably this is probably a Sanger sequence that had a weird thing at that position. Actually, we can check and see if it was Sanger.
Like
It
the one that it's going against. I see.
yeah, this is probably Sanger. Oh yeah, it's from 2017. This is definitely a Sanger sequence because this is
That
from
is
before Steven started really doing nanopore.
Is that
So,
22 or 23 or 22 or I
oh
don't
what
remember
I
when he
don't
started doing nano.
I don't know off the top of my head. 22 maybe. Um
Okay.
so this looks like it has really good consensus. Like this might be one I should I'm less skeptical with Amanitas than I am with some other groups because Amanita people are slightly psychotic and um mistakes don't really perpetuate that much because there's a bunch of people chiming in.
Okay.
Does that make sense? Like there's some groups that are a lot more confusing, and then there's some groups like Amanita where like there is a lot of people that care a lot about the group, and so there's just been a lot of work done. So like Amanita is definitely not one that I have any super concerns about it. And like Solaniolans, I'm pretty sure is an American species, which lends at like specifically an East Coast American species, which lends me can't spell. It it tells me that I don't really have to go digging to see if it's like some old European name that is getting misapplied. Yeah, this is a
Oh, yeah, 'cause
so it's
there's
Nova
that.
yeah. It's the Nova Scotia type locality is Nova Scotia, right? And it's a newer species. Like newer species tend to have better concepts, I find. The really confusing stuff is like the 1700s like freeze species where like there's like 15 different things being called the same things and when people finally go sequence them like everyone calls these disparate sequences the
Right.
same. So, okay.
So something else that you're looking at, even though you have all these 100% matches, is to ensure that the name is not being misapplied if you were to
Yeah.
use it for that particular species.
Yes.
Now, what if everything that was similar was in Europe and there were no American, like, sequences in the Americas at all? What would you?
That does not necessarily mean that it's not the same species, right? Um
Sure.
if so let's just assume let's scenario is maybe we have a type specimen that's from Europe, right?
Uh-huh.
So we have a type, it's a recent paper, we know this is the species, like we know this is what this represents, and then it pops up in America. That probably
Sure.
just means the distribution of the species is more than they realized in the initial paper, right? We
Absolutely.
it's hard to make positive claims about like the range of a species. Like it's hard to say this species occurs in this range but not in other places, right? Saying it's
Yes.
not in other places is not usually falsifiable, right?
Okay.
Unless a lot of research has
That's
been done. So there's very
sure.
few times when I'm going to say like this species definitely doesn't occur in the United States. Like basically the only times I end up saying that are for stuff like Macrolepiota procera, where there's been a bunch of sequencing done, and like we haven't found a single Macrolepiota procera in the United States, or like Cantharellus sabarius is another one. Like the American chanterelles, from whatever people have saying, or what from what everything that people have been able to find, seem to be distinct from European Cantharellus sabarius. And like same
Okay.
thing goes with some rustula species and stuff, right? But if it's like a lesser reported species,
Mhm.
there's just a chance no one has found it or ID'd it because they assumed it wouldn't be here. There's just really weird disparity when you look at stuff on iNaturalist for certain fungal species, because some
Oh
species
yeah.
will have millions of observations, right? So like something like, let's just
Yeah.
do Macrolepiota procera because I was talking about it, right?
Sure.
So this is the range map for Macrolepiota procera.
I see. Okay.
The only reason there's no Macrolepiota procera observations in the United States is because I fixed them all, but up until Massalenta and Pallida got published by Stephen, like there was a bunch, right? And there were people that would go through and be like, look, this species still needs described. We haven't found any evidence of this species in the United States, and no, there hasn't been any DNA evidence of it. But it's hard to fight back against INAT when there's not like an actual species. These down here in like these all in South America are probably one of the South American species of Macrolepiota. I haven't been able to find good enough source information on that to really like dig in and figure out what the macro differences are between them. So I haven't really worked on that. And then like there's some Macrolepiota species from like Southeast Asia that are also these are also probably not technically Macrolepiota procera, right? But because that's what everyone knows as the parasol mushroom, like every guide is going to call um a parasol as Macrolepiota procera. I'm trying to find a book. Sorry, give me a second.
Yeah, sure.
Uh is it here? I'm trying to find
Mm.
my recent because I got the recent Peterson Field Guide. It might be in a different room. I'll be right back.
Yeah.
Okay, so my books are everywhere. I need to figure out where they are. But so this is Appalachian Mushrooms of West Virginia and the Central Appalachians. This is published in 2003, right? So it's 21 years old. And this is actually a pretty good book. Like this is a solid book for the region. But I guarantee you when I go look up Macrolepiota, it's still going to say Macrolepiota procera, right? Because this is a 21-year-old book.
So there were a bunch of observations because of the morphology, but then once you did the sequencing, you
Mm-hmm.
realized that it wasn't actually the same species. Is
Yep,
that true?
basically there's yeah, so 72. So basically like there's definitely some morphological similarities. There are some morphological differences, but like there sometimes what happens is you get like these species that are just like dump species. Um
Okay. Yeah.
and so that'll be the species that ends up in all the field guides, and so that's what everybody wants to call it. Yeah.
Okay.
So Macrolepiota procera, right? So again, 20-year-old book, but I guarantee if I pulled up the Peterson Field Guide, that
Mhm.
I don't know where my Audubon book is. Oh wait, I have it right here. So I guarantee if I pull up the Peterson Field Guide, it would probably also say that because the Peterson Field Guide isn't super up to date on names, even though it was published like last year. Um, but basically, people get guides like this that are a little bit out of date because these books are out. These books are out of date the minute they're published. Every single one of them, because they just don't move
Sure.
as fast as like scientific data. But people
Absolutely.
take them as gospel.
Absolutely. The
um,
half-life of science.
basically.
Oh,
And that's okay. That's how it works. And like there's not really any
it is.
harm in IDing Macrolepiota macilenta in the eastern United States as Macrolepiota procera, but it's technically not correct, right? This is a very weird tangent I'm going on, I'm sorry. Here we go.
Oh
So
no, this is all good information.
so this book is the new Audubon book that came out last year, I want to say. Came out in 2023, I believe. What page am I on? Macrolepiota 75. So, yeah. So, it's a little better. It's called Macrolepiota procera group, right? So,
Oh,
I don't know if you
okay.
can see it, but it says Macrolepiota procera group up here instead of just Macrolepiota procera. So, this got published before Macilenta and Pallida were described,
Interesting.
right? So, the person who Jacob Polk did a lot of the taxonomy in this book, and he's a very good, he's knowledgeable, he knows what he's doing. It's just that
Yeah.
information wasn't out yet. So even though this book is very recent, it's out of date. Right?
Especially with fungi, I would think.
Mhm. So if someone
Yeah.
buys this up-to-date brand new shiny Audubon Society Mushrooms in North America book and
Uh,
uses this to ID their stuff, they're going to be wrong. And that's okay. I think this tangent started because I was talking about falsifiability and it just went on from there. But
I also ask a lot of questions that are off
point
topic.
being, it's hard to make exclusionary determinations about range unless there's been a very wide sampling of stuff. So I've definitely
Oh,
found stuff from recent papers that we find one that's in like Europe, and then I find one in America. I have a good example of one from mine. It's this one. So this is the I think this is like the only observation of this species on INAT. Um, so
cool.
it came back as match. I think it was like one base pair off the type for Lio. Oh, there's two now.
Oh,
Apparently, we found another match in Ohio. Maybe. Wait, where's the other one?
where'd you go?
Oh, there's one from 2 years ago. Okay, that's weird, but okay. So we're gonna ignore that one because it's not really. I don't really see any justification for that.
Yeah.
But so I look at the paper for this because it came back matching the type specimen for the species. It was only described a few years ago. It's not very old. And there was
Oh, okay.
also, so it was described from Turkey. There is another paper that reported the species from Austria. And then I found it under a non-native Norway spruce, like a landscape Norway spruce, right? So
Okay.
this species
Huh.
seems to associate with like Asian, you know, European Asian spruce species, right? So like one paper reported under Picea orientalis, another paper reported under Picea abies, which is Norway spruce, right? So even though it's way out of range, the substrate
Hmm.
matches.
Absolutely.
So it's not unreasonable for me to call this Liophyllum turcicum. I could
Interesting.
potentially go through and do micro if I really wanted to on this one and like, try to figure out further, but I don't think I've gotten the specimen back yet, so I haven't done that. But it's for now, it seems safe, right?
When you say do micro, are you talking about actually looking at it like
Hmm.
microscopy sort of
Yep.
stuff? Okay. You would
I'm
look at like, the spores, or what would you look at under the microscope?
spore size, like if there's any cystidia, like how many sterigmata does the basidia have? Like, so does it, so basidia. This is turning away from a validation tutorial
We
and
can do
into a.
whatever.
That's fine. Honestly, if it helps, it helps, right?
Okay.
So
Yeah. No, I'm good with this. This is great.
this is a really good page for microscopy terms, like so
Ooh,
Michael
I have that.
Quillon, right? So he kind of just talks about terms and stuff, right? He's got like this is kind of what spores can be, and like these are the different types of spores you might have, or this is how you
Yeah.
create a French aqueduct section. So you might look at the structures in the hymenium, like the pileus, what have you. Um, so like these are going to be basidia and cystidia. So the basidia are going to have these little guys that are the sterigmata. Usually there's four. Some species might have two, some species might have eight, but so if it has something other than four, like that's going to be fairly diagnostic. Like Amanita bisporigera is called Amanita bisporigera because it only has two sterigmata on the basidia. There's only two spores per basidia,
All right.
right?
Cool.
Um,
Yeah.
the
Hmm.
cystidia are just like these little structures that are on the gill edges or the pileus or like there's various places on the mushroom cystidia it can appear. And they might be called like pleurocystidia or chylоcystidia or what have you, right? There's different terms. Um, and so you can look at these, and the cystidia shape will kind of help you tell what species you have. And there's like other things you can look at. I'm not the best at micro, but I'm learning. Um,
Cool. Yeah, that's awesome.
so,
That's really, really
for example, this is not one I don't think I've gotten a sequence back on yet, but a species, Strobilurus esquilentus, which no one has confirmed. All right. So the range map on this one, I want to say before I found this, yeah, my observations are the only Ohio observations of this species, right?
cool.
I haven't actually
That's
gotten the
fun.
sequences back on these yet, but I did the micro. And
Oh,
so this is
cool.
the cystidia of Strobilurus esquilentus, right? And there's actually a fairly good Strobilurus monograph that came out a couple years ago that goes into like stuff like substrate specificity and the cystidia shape. And for Strobilurus, cystidia shape is very distinctive and that kind of a if you know what your substrate is and you know what your cystidia are, you can pretty much pin it to a Strobilurus, even though macroscopically they all basically look the same. So this is
Cool.
growing underneath a planted Norway spruce, which is the preferred substrate for Strobilurus esquilentus, and I can look at the cystidia and know I for sure have Strobilurus esquilentus, right? Matches the spores, matches the cystidia. It is what it is. Which, if you're going to invalidating sequences, Strobilurus in general does not sequence with ITS well. A lot of them have the same ITS barcode, so you need to know
Okay.
substrate for Strobilurus. So
Uh,
if you're looking at a Strobilurus sequence and you see it's on Norway spruce, you
okay.
can confidently say it's Strobilurus esquilentus. Um, that I put some of that in the problem groups document too, by the way. So it is in that document.
Oh, okay.
Uh,
Did y'all send that to us or how do we access that?
I think it's in the Slack. I think you'll get access to it when
Oh, okay.
yeah. If you
Great.
don't, just let me know. And I'll let you know.
That's
And
good.
I'll get it to you. Okay, let's
Um,
do a little bit of validation. Okay, so this is
I'll try
definitely
not.
Amanita solaniolens. So this is one of those cases where I'm not going to be super specific because like we know for sure this is an American species. It's from the right environment. It looks correct. It's got a ridiculous amount of 100% matches. Like this is not one I have to be overly skeptical with.
Certainly.
So I'm just gonna kind of copy like two. I say lots of 100% matches, including that and da-da, and then I'm done, right? It's not one that I have to spend a lot. I don't have to go into the weeds on this one. It's just not worth it.
Certainly.
There we go. I might publish a book someday, The Mind. We'll see. Give me a few more years.
That'd be awesome.
Validated. Amanita. What did I say it was? Solaniolens. Solaniolens. It is from Mass. I will copy my notes over. And this is one I'm going to have to put this name in the provisional observation field just so it'll pull over to Micromap properly. Plenty of
Okay.
matches. No. Notable. No. Gem make status is going to be ready. Sequence correction. I did not. I haven't found this one, but apparently it smells like potatoes.
I love
thus,
that.
thus the name.
Old potatoes.
The old potato amanita. You'll see me add stuff to a few more projects than we tell people to sometimes.
Certainly.
But cool.
Once you know more and
Mm-hmm.
know where these things are desired, that
Yeah.
makes sense. Yeah.
Who is texting me? My mother. Let's give it maybe another half hour and then I gotta go get pizza.
Yeah, no worries.
Hmm.
Whenever you need to go, I'm good. I'm easy.
Yeah, you're fine.
I appreciate your time. Thank you.
Hmm.
Thank you very
I
much.
appreciate you. I'm just glad you're communicative
Yay.
because, like, I would much rather do a stream with you here, like, offering helpful commentary because otherwise I feel like I'm talking at a wall and it makes me really anxious.
Yeah, yeah. I can understand that, especially when you're doing it for so long and you're like, I've been doing the same thing, like, and now what do I say? Just repeat everything I've just done already?
Right?
Like,
Like, is it useful? So something I will do
yeah.
for OMDL is we have these OMDL numbers. Not everyone does this
Mhm.
consistently, but I will usually go in. I have a field for them, and I'll usually go
I
in and add them.
remember seeing that.
Yeah.
I wanted to do that last night and I was like, wait, we're not an OMDL.
Yeah, I
So I
mean,
didn't
I
even say,
don't know if I mean, you can. Like, if you're looking at OMDL sequences, you totally can. That's fine. This is just what we
certainly.
use for, like, our internal lab code, basically. Um,
Right.
so let's see, that's GenBank ready, so that's validated. So let's go back to that one. So now I'm going to go back to that court that I blasted bookmarked and see what's going on with it.
Cool, cool, cool.
Apparently I need to update the species. It's weirding me out that Steven added this update because, like,
Oh, that one.
it wasn't there before.
Nice. That's the number of nucleotides.
It's just how long the sequence is, which is not a
Nice.
bad thing because occasionally you'll get chimeric sequences that are like really long,
Right.
and sometimes those are not good.
Remember that was something that Hart mentioned in the
Hmm.
training somewhere.
This is
Which,
kind of a weird one. Okay, so it's got 100% match to this one. Which is one of Shannon's observations. So Sulcatus is a Cortinarius expert. Like, she's very good at
oh,
Cortinarius. So this might actually be one case where I'm going to put a bookmark on this and ask her if she has a temp code for this one because she might already have one that she hasn't put up on iNet. I don't
oh,
normally
really?
do that, but like
Okay.
there are a couple people like anything
Mhm.
in like the Pacific Northwest, like a lot of that stuff Danny has temp codes for already. So if you make a new temp code for it, he'll then pop in and be like, "Oh, it's this one," and then you just made a
Uh,
temp code for no reason.
uh,
And when it
I
comes to
see.
stuff like that, I'm just gonna defer to him because there's like a lot of mycologists in the Pacific Northwest that have like their name plus a temp code. So you'll see a lot of Russula with like the harrower and then a number, and like harrower is a Russula studier basically.
Okay.
So it's just not worth getting weird about. So
I see.
yeah,
Are these people doing their own sequencing or they just know so much that they know it's
a lot
new
of people
and
are
good?
doing their own sequencing. Like a lot of it's like academics that are doing. Uh,
Cool.
like academics that are doing like what do you say? Yeah, like their own Sanger. I mean, you could do your own sequencing.
Sure. Are they doing Singer?
Hmm. Sanger or
You
nanopore,
or okay.
probably. This is probably not the same species. Quartz are like so
Cool.
quartz are one of those groups where like a couple nucleotide difference is a big deal.
Oh,
So
really? That's
yeah,
the way.
so the fact that there's
Yeah.
four, there's one, two, three differences that are solid. This Y resolves. This T resolves because it's a hollow polymer repeat, but this one does not. So there's three real differences in a gap. This is probably a different species. Quartz are tight, usually, but it does match this observation of Shannon's. So what I'm going to do in this case is I'm going to actually do this. I don't know what section this is in. I'm just gonna put it. I don't know. I don't think it's defibulati. I don't know Cortinarius sections well enough to figure out what one it is. So let me just look and see what's going on here. Yeah, this is. Yeah, so even up here, it's not particularly close, right? Quartz are weird like that. Like the difference between Cortinarius tursii and Cortinarius croceus is like 2 base pairs.
Wow.
Yep.
Okay.
Hmm.
So the species generally, do they not have any differences or generally or
What, like quartz?
yeah,
I mean, they'll have
yeah,
like one or
quartz.
two usually. It's just, I just there's just a really the ITS region for them doesn't seem to evolve very quickly, so there just doesn't seem to be a lot of differences. Hemaloma are even worse. Like Hemaloma, a lot of Hemaloma you can't distinguish just based on ITS. Um,
Oh, weren't we talking about that last night? I think
we were.
something. Yeah,
Yeah.
cool, cool. Nice.
Okay, so I tagged Shannon and I'm gonna see what she says. So I'm just gonna leave that book, come back. And then I will just bookmark that and come back to it later.
Oh, are you really keeping track of notifications on a naturalist then?
Oh yeah.
Like pretty
Hmm.
okay.
And like even so, if I go back and like review my stuff too, that's why I like leave it as like comeback and flags and stuff because then when I go and look at the sheet again, I can check my comebacks. So I've got a couple on like my fundus plates right now that before I upload them to GenBank, I need to resolve some flags I left on some of my stuff because they were problems. Like there was one that was like maybe a hybrid that we were trying to figure out how to notate the name of because we found a couple that might be hybrids and uh we were trying to like when like the two haplotypes blast to wildly different temp codes, it's like okay, what do we even call this? So we're trying to figure out if we should just like upload them to GenBank as like a temp code hybrid notation or something. I don't.
Wow.
It's
That's
not
wild.
something you would have caught with Sanger because of the way Sanger chromatograms work, but like with Nanopore, because it's a bunch of individual reads, you can actually kind of go in and pull out haplotypes a little easier.
Oh, that
Hmm.
makes sense.
Yeah. Because like a Sanger
Hmm.
chromatogram just looks like a bunch of peaks and valleys. Like
Sure.
I can, yeah, like this is what it looks like.
Right, yeah. I've seen some of
So
them on Steven's
like it's a little
videos
harder to figure out if you have a like genuinely a two like a multi haplotype situation going on.
for sure. That's really cool that you
Hmm.
can do all this
Yeah.
fun stuff.
Right? That's kind of why I like looking at this
Yeah,
stuff because it's
sure.
just fun. I don't know.
Sure. No, it's fascinating.
Well, you are also a
It's
data nerd, so that tracks. That tracks why you
right.
would enjoy this.
Oh yeah.
All right. Sorry, my mom keeps messaging me. All right. That needs reblasted. So sometimes this happens. It doesn't pull up the local blast. It just pulls up
Oh,
the NCBI blast results. I'm just going to
okay.
take a glance at the. Okay, so this is the reverse. You can tell it's the reverse because there's a little negative under subject cover.
Yeah.
But
Hmm.
why is this matching both Beningiana and what the heck? This should not. Sorry. Okay, I don't think this sequence should be here, but I'm just going to leave it, I guess. Um, so lots of 100% matches. You know me. I'm still gonna go and check the. I'm gonna go look at his genes. I'm gonna look at those. I'm gonna
Yeah.
peek under those nucleotides. See what he's got going on under his skirt. It's probably not gonna be um that weird, but it's still worth checking. But see, I like checking these, right? Because like, so you know how I was saying like Croceus and Cortinarius croceus and Cortinarius tursii are like 2 base pair difference, right? Normally, they seem to have fairly stable ITS, right? There's not a lot of ambiguities, but I found one that had ambiguities in the exact right spots to resolve it, so it's 100% match to both the type specimen for Crotonarius croceus and the type specimen for Crotonarius tertii, and so we're just like, okay, is this a hybrid? What's going on? So
So what do you do with that?
exactly like that's
Oh, is
it's
that the one you were just talking
that's
about?
one of
Like
the
trying to
that's
figure
one of the ones, yeah, like
out one of
it's
them.
one of them. There's a couple. uh
Wow.
so and this is like way more in the weeds than you're probably going to want to do once you start like doing this at
For
first.
now,
Like this
yes.
is this is like the progressive evolution of me getting more and more and more in the weeds on this stuff until I just apparently decided to go full bent on being a little bit crazy. Crazy pants, crazy bananas.
How long have you done validation?
A little over a year at this point.
Oh wow, so not too terribly long.
Yeah, I think I started
That's
up
awesome.
like end of like fall last year.
Oh
Maybe
cool.
before.
Wow.
Well, actually, wait. What? No, it's been a little longer than that. It's been like maybe 2 years at this point. Sorry, time doesn't have meaning anymore.
Sure, you're good.
Like
Yeah, time's
I'm in my 30s. I time is meaningless. So yeah, like 2 years at this point
I hear
probably.
you.
Maybe a little less. I don't know what
Cool.
month it is half the time. Okay, so I'm not pulling out anything obvious, so I'm just gonna close that. So this is definitely this blusher. I'm not even again. This is one I'm like not even gonna question it. It's a provisional. It's one of Rod Tullis's provisionals. Rod Tullis's provisionals are solid, right? So we're just gonna put it in here. So when you want to get to complexes, I always forget this, but yeah, if you type in like the name, it'll pop up with that. So I'll show you a better one.
Mhm.
So if you type in the species and if there's a complex attached to that species, it'll pull up the actual species, but it'll also pull up the complex. So when you're doing these, like look and see if there's a complex. hmm
Oh, that's really good to keep in mind. All
hmm
these nuances, man.
Yep. And this is right. There's just a
Ooh.
lot of nuances.
Ooh, yeah, I think I've probably done some of my iNaturalist observations wrong at this point then.
It's not a big
But
deal. Like the beauty of
cer-
iNaturalist is that you can just go back and correct stuff.
certainly,
Like I've
certainly.
definitely
And I guess,
no,
oh,
go on.
what?
Go on.
I was gonna say, I guess if I put it in the complex, then it isn't wrong. It's just not as narrow as it could be,
Correct.
maybe.
Yeah. And it won't hit research grade at complex unless you hit the so at the very bottom there's data quality assessment. So if something's at
Uh-huh.
genus level or below and there's consensus but you can't get it to species, you can
Yeah.
click based on this evidence can the community taxon still be confirmed or improved and hit no, it's as good as it can be, and then it'll be research grade at genus, subgenus, section, whatever.
Really?
I don't use it that much. Like most most of what I use it for is like if I'm just doing general identification on INAT and someone posts a shitty picture of a red russula from the top, it's not getting past that. I'll go check this. And that way it'll knock it out of the identification queue cuz otherwise
Oh.
cuz otherwise it'll just be it'll not be ever research grade, and then every time someone goes through and validates it, they'll just have to add add their opinion or skip past it, and then you'll end up with these like garbage observations that have like 15 agreeing people agreeing that it's a russula, and it's like
Oh, wow. Man, I'm
mm.
learning so much, Jessica.
I'm
So
glad.
much.
I'm glad.
Wow. That is so cool. Ah.
Yeah, there's like little things you pick up over time.
Oh,
um
certainly. Tribal
s-
knowledge is, I guess that's probably not the best thing to say, though, is it? Hmm. There's probably better words for that. I don't know.
no, there actually is a word for this, but I can't. I i don't know, something.
Yeah,
There
definitely.
actually is a phenomenon, I can't remember the name of it, where like
Uh-huh.
husbands and like people who are in relationships, like long term like situations, like one person will remember the things, right? Like the wife or the husband, like someone will be the person that has like all the addresses memorized, and every time the husband like needs to recall it, they'll just be like, "Honey, what was that address?" Right? And
Interesting.
I swear I read an article a while ago that was talking about how like people like in our generation use Google for the same thing or use Wikipedia. I do it. Like I can't remember stuff sometimes and I just like look it up on Wikipedia. I can't believe more
Oh,
people
sure.
don't have this instinct. Like
Yeah.
if I don't know the answer to something, I go look it up because this has access to more information than anything anybody at any other point in history. And like you do have to kind
Yep.
of know how to parse out what bad information is versus good information, but like if you can do that, you can
Yep.
you can educate yourself on any topic.
It's incredible. You really can.
And this is why I get so frustrated about like just mass ignorance because there's no excuse.
Oh my gosh. Well, when people are trying to feed people lies to stay in power, like they do a really, really, really fantastic job of that.
And and yeah,
But
and I don't really blame like the people being manipulated because the only like bulwark against misinformation and propaganda is to be educated because like everyone's subject to some propaganda. God knows I am. It's just
oh, absolutely.
I at least attempt to examine my biases and like not be afraid
Yeah.
to change my opinion if better information comes along. And
Yep.
I'm not going to get
Yep.
defensive if someone says something and corrects me. I want to learn new
No,
information.
I know, right? One of my things that I used to tell everybody is that I love to be wrong because like you learn so much. You like start
Mhm.
reorienting your
Yeah.
whole like so much around it. But
It's a
that's just not
mindset. Yeah. It's a
it's a
mindset that you have to
total
cultivate.
mindset. Yeah.
And
Yep.
unfortunately, it's just there are certain groups that are motivated to not cultivate that skepticism because it's easier to control people when they're not skeptical.
It's there, yeah, it very much is.
And like when I say skepticism, I mean a very specific type of skepticism. Like I don't mean just question stuff and then not do anything about it. Question stuff and then find the answer to your question.
Yeah. And it can be hard too because when you're like trying to question things like on Google or something, it's going to feed you back
Mhm.
your biases at the same time. And so
Yep.
you're like,
Oh, it's getting worse. Like it's Google has become a lot harder to actually find good information on.
it really, really is. Yeah. That's why I try to use Ecosia as like a first thing, but man, it's hard because it just doesn't work as well for
Yeah,
me.
I might sometimes I use DuckDuckGo. I might switch to Ecosia because like sometimes I need to get past like whatever AI generated garbage that Google is trying to feed me. So I might try out Ecosia because I was not aware of it.
Yeah, they plant trees. They're great. I
I
think they're
like it.
great. If you find out they're not great, please let me
I
know, but I
haven't
think they're great.
I haven't heard anything about them. So,
I don't remember where I probably like I delved into like climate like understanding about a year ago
mm.
and somehow came across it during my fellowship, I think.
Also something I just like to do is I like to find like educational content or creators or what have you that are experts in their field and are willing to share their knowledge because they exist, right? There is a Discord I'm in. I don't want to pull out my whole Discord on stream. There's a Discord I'm in. I don't live in Florida or the tropics, but I'm in the tropical weather Discord because I still like keeping track of this, right? And the tropical weather Discord has actual meteorologists that post, like people who work for NOAA and fly the hurricane hunters post in this Discord,
Oh wow.
right?
Yeah.
So the people
Need this.
analyzing the data on it are knowledgeable, experienced, credentialed experts in the field. And so being able to like go there and see what people are saying there helps
Yes.
inure myself against like all the misinformation that was happening about the hurricane.
Absolutely.
right? Same
That's
thing with
yeah.
other stuff. Like I follow on YouTube. I mean, aside those two people, like Clint's Reptiles, he's like a PhD biologist. Like I said, Forrest Falkai is a bioanthropologist and is like got like one master's and is going for another one, I think. There's another one I follow who is a primatologist. Like she's going for a PAT in primatology. There's an archaeologist I follow. There's uh just, science educators and knowledgeable people. Like I've got historians that I follow that have like do university lectures and will just post like an audio recording of their university lecture on YouTube.
Well, you with all the work that you're doing and all the stuff that you're starting to publish online, you'd be like one of those people that's like the validator.
We'll see.
So,
I
like
I
that's pretty
I
dope.
we'll see. We'll see. I don't know if I'm going viral with fungal fungal validation. Okay, this is Trimides hirsuta, and I
Well,
don't even know.
viral doesn't necessarily need to happen, just reaching the folks
That's
that
true.
need the information.
Hmm.
Like, just me watching, I was like, wow, this is just incredible. So it's you're doing really great work. So
Yeah.
that's all. You're talking about all these other people, and I'm like, look at yourself.
I have self-esteem issues. I guess I'll look at the genes for this one. This is Trimides hirsuta. I might
I
not
am
even.
his
There's a strong chance this is one I'm not going to be too skeptical about because it's freaking Trimides hirsuta. I mean, I just want to be helpful to people.
Yeah, it's obvious. And it's obvious and wonderful.
I like info
It's
dumping.
so, so
it's
yeah.
like it's like the nerdy info dumping. I just want to find people who want me to info dump at them.
I love it.
I'm like, there's facts in my head and I want to share them.
Absolutely. No, I totally get you on that. I love sharing. I'm gonna say hi to my kitty who's meowing in the other room.
Okay, I guess this is ambiguous. Yeah, I would call that ambiguous. It's friggin' editing a Trimides hirsuta sequence. What's wrong with me?
Is that not a common thing?
It's just a really common species.
Trimides in general? Is that
Trimides as
or
a
no?
genus in general is just they're everywhere.
Okay.
Um, see how fast I am when I'm like just doing it. So I'm gonna just do a new blast. I guess I'll really don't need to do a new blast for that one. Let me see if I can find one that actually has a local blast. I'm gonna possibly open a can of worms that I don't want to open. We'll see. We'll see if I opened a can of worms that I didn't want to open. Nope.
What you doing?
So I'll do this sometimes. Blast it against like that species name
Okay.
just to see if like there's good consensus in GenBank on what is being, what the species represents. It's everything is pretty solidly a good score. Like the scores are high. Like the lowest is like 98%, 8.5% similar.
Is there a particular reason why you decided to do this or just for
Because I'm a masochist, and if it was different, I want it would. I because I questioned it, and if I question something, I have a pathological need to like go look up the answer. It's
Sure. No, that's a legit reason.
um yeah. See, and Steven probably was going through this and saw that it was Trimides Caserta, and then he just added his ID because he was probably going through a bunch of Trimides Hirsuta. Yeah, there's. I don't usually check everyone to see if like it's got good consensus. Like some things I know they're gonna be fine. Like but I, I don't know, man. I just don't think I've checked one of those in a while, and I just wanted to make sure I wasn't misremembering. Because I have found
Right.
like errors perpetuated in the system that weren't caught.
Absolutely.
Did not export. Come on. I keep getting like Republican spam texts.
Oh, really? I haven't gotten any of those.
Someone
Ooh.
someone named Roland apparently used my phone number to sign up for some Republican like
No.
text list, and I keep getting like NRA messages. So that's it all gets like shunted to my spam, but I was bored earlier and looking through my spam and it was just like, "Oh, okay."
Oh my. Ooh.
I honestly waste your time phone banking on me. Go ahead, do it. Why
Right?
why is this not
Please.
updating? I don't want to have to manually transfer this over. Yeah, like I was for a while there, I was like not blocking some of the worst political posts because I'm like on like YouTube, like the political ads on YouTube and stuff. Because I was like, sure, waste your money on me, but I had to block them eventually because they're too much. They're too I. It's too much hate speech. I can't.
It is. I went the other day, I was like, why is this crap showing up on the side of Jessica's YouTube video? This is, and then I just like scrolled and like was like
Well, you're in Texas
reporting
too, so
them.
you're probably getting as bad of as what we're getting.
It's gross.
Yeah, it's
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 sad.
really gross. Like
It's real sad.
our Democratic Senator Sherrod Brown is one of the most contested like Senate seats in this election right now, so it's just been a like constant assault. And the ads are some of the most like bigoted, transphobic, nightmarish garbage that I like.
Yeah. Yeah. So it's a
I am too empathetic. I just can't.
I know,
I
dear.
can't. I can't
I
with
know.
it.
I know. I feel you.
So,
It's rough. It's real
oh
rough.
wait, did I put the wrong? No, I didn't. Yeah.
Oh,
So sorry, not trying to get on a politics tangent here.
I mean, right now is that time to do that, isn't it? It's just
True.
like behind your shoulder and it's gonna be here. But yeah, we have to
Yeah.
try to
I'm trying to
not
like basically I'm just trying to compartmentalize and not like lose my
yep,
mind.
yep. I feel you on that. It's easy to lose your mind.
Go a little nuts, yeah. Let's do one more. Ooh, what are you? Apparently it's a Foliotina. Thanks, Steven.
Where did you see
Steven
Leotina?
commented.
Oh, right there. Oh, he commented.
Hmm.
I see. I see.
That's
How did
okay.
he know that?
Because
Just
he
from
he validated something that was under that same temp code. So Steven will just every time he like goes in and like sees other stuff that hasn't been validated yet, he'll just chime in. Usually, it's just what he does. Let's do one more so we know it's probably good.
had it been sequenced yet, or was
Yeah.
it just like a morphology? Okay, cool.
Nope. Yeah, it's been Steven doesn't
It
do
was
morphological identifications.
I know I was like, that sounds a little
Nah.
interesting. But
Steven doesn't do that. Steven also doesn't check his INAT notifications, so if you ever tag him on there, he won't see it.
okay,
If
that's
you
good to
if
know.
you need
Thanks.
information from Steven, just message Steven on Facebook.
Right, right, absolutely. I found that out.
Yeah.
It's definitely the easiest way.
Yes. He is And he responds. It's just funny because there's a couple people that like have nightmarish INAT inboxes, and yeah,
Um,
I mine occasionally. I'll sometimes come back from a long weekend, and
mm,
oh yeah, okay, I'll come back from a long weekend, and there will be just hundreds of notifications in my inbox, and that's always a nightmare to clean out because like INET doesn't have a good way to retrieve like old notifications. So if you accidentally like close out of like so if you like pull up this little drop down window up here and then I close out of this before I had a chance to like look at it,
mhm.
if there was a lot of them, they'd disappear. Why are these all failing? Hmm. Whatever. Uh, wait, I did that. I did that. That consensus is good. So we're gonna copy this over. Yeah. So the way Steven does it, Steven just like looks and sees like, okay, there's not any big jumps between like specimens here. So he doesn't usually do sequence
Okay.
editing unless it's like something egregious. Um,
Uh, okay.
but I do because I am psychotic. um,
You want to be thorough.
we have different validation philosophies, which is
Sure.
fine. It,
Yeah,
honestly, the more different viewpoints we have on stuff like this is the better. Um,
absolutely.
but if I actually, let's just do this. I'll show you what's going to happen. So what's going to happen is those ambiguous nucleotides that I put in there was probably going to end up resolving. Yeah, because see, that one matches to that, so that's a Y. So CT is a Y. And then for
Okay.
these two, so these were also ones I think it picked up. Oh, it didn't pick up that one.
Mm.
So that's solid. So it doesn't pick up this one, but it picks up this one.
The ratio or the percentage just not there to pick up the other one.
Well, it was a solid T. It definitely wasn't. But that
It was a
doesn't.
solid D.
But there's a chance that this one could have like an ambiguity there that just wasn't done. But either way, like so, all these ones that are 98.6 similar are going to end up being 100% adjusted matches once I reblast that. Oh my gosh, what did I do? Once I reblast it. So I'm
Okay.
like not going to wait for this blast to finish. I'm just gonna grab a
Sure.
couple. And this is the nice thing about the temp codes is like you don't really have to be as critical about like where this name comes from because it's not an official published name. hmm
Oh, that makes sense.
Because all the temp codes are saying is this is a bunch of sequences that are all the same. It's not saying
Sure.
this is where this. It's with the published species you have to be a little more critical. Um,
You ever see these temp codes resolving into something
mhm.
that is published?
Yep,
Okay.
yep, it's definitely happened a couple times. We
Cool.
actually so the type sequence for Russula parvovirusens hadn't been published. So Bart Buke, who is the describer, hadn't actually uploaded it to GenBank yet, and we had so many Russula temp codes, and we were trying to figure out what the actual like parvovirusens like code was, and it was a fairly recently described species. So I just like emailed Bart on a whim and was like, "Hey, we're doing this sequencing project in the U.S. Do you happen to have the sequence for your holotype for this?" And he's like, "Oh yeah, here it is." So
Perfect.
so it enabled us to go. This temp code is parvovirusens. Yeah,
Interesting.
ITS sequence matches the holotype. Yeah.
Cool.
So like that's one temp code that has been gone.
Huh.
There was a pseudohydnum gelatinosum temp code that we had, and then someone published a paper and described a subspecies. That's what is our pseudohydnum gelatinosum species in America. So that temp code went to that subspecies. It has happened. It does happen. You know.
Is that
Hopefully,
something ever y'all do,
yeah. We've
or
definitely
is it
we've published a few things so far.
Right.
And that, I mean, we were talking about the last meeting, we were talking about publishing some more stuff.
Nice.
Oop, I probably should. I probably should actually do the validation correctly instead of just getting distracted.
Sorry.
You're fine. I'm the one who's talking. What's the sequence validator? Why is... Okay, I don't need that anymore. Why is this not exporting properly? Please do that. Thank you. There we go. So since this is an Ohio record, I'm going to add it to the Ohio project too. There we go. That's all good. That's good. All right, I'm gonna go get dinner. Did you have any more questions? Do we just want to like chat again at some other time?
Let's chat again another time. This
Okay.
was fun. Thank you so
Yeah,
much.
of course.
Yeah. Fun and informative. Like, woof,
Yay! Glad to
perfect.
glad to be of service.
Oh, well, you
Uh,
have a lovely dinner. I
you,
hope it's nice outside. It's nice here.
it's been kind of cold and damp, but that means there'll be some mushrooms when we go out this weekend, so I'm not that upset about it.
Fair enough.
So,
It's been in the 90s and we have a 70-degree day today. So I'm like,
for Texas, that's like, well, it's been so dry and it's been raining for like 3 days straight, which is so good.
really? Oh,
So,
nice.
I'm gonna
We need
go.
some too.
Yeah, there's an old growth I want to go look at this weekend and see if I
Oh,
can find a couple. There's some weird stuff we found about this time last year, so I'm hoping that I'll find it again because there's a hygrophorus that I found last year that ended up being fairly novel, and I would love to find a fresh specimen because it's nicer to do micro on fresh specimens. Like rehydrated specimens kind of suck to micro,
that makes sense.
but
Y'all have old growth up there too?
a
I
few.
didn't.
So
Really- That's cool.
it's Ohio doesn't
We're
have a
looking forward
lot
to
of
it.
old growth forests, but we do have a couple.
Yeah.
Yeah.
We're trying to find, we're looking for a place to move, and I'm just trying to like think of all these different things. So old growth is something that like draws my heart like
Yeah,
tremendously.
well, here, let me stop the stream real quick. Here we go.